Biology is so broad and diverse. If you restrict yourself to studying the standard organisms, you miss a lot of interesting phenomena. So I think it was actually a brilliant move to go outside of the normal range. The revolutions come from giving up on some fixed idea that everyone holds and realizing that actually things could work in a different way. And that's happened again and again and again. It's the advances in how you think about the problem that really lead to new developments. It's such a fun and interesting field, right? We're always finding new things. It's just so exciting and interesting, and it's really a great privilege to be able to work in this field and really study something so fundamental.
In this episode of the 632 Nanometer podcast, Misha, Xinghui, and Mike travel to Chicago to take another bite at the origin of life apple. Nobel laureate and previously Harvard, now Chicago professor Jack Szostak is their second guest in the Origin of Life series of talks with scientists working at the cutting edge of one of the most profound unanswered questions. How does matter become living nanotechnology, and how did it happen here on Earth? As they enjoy the bird's-eye view of Chicago from Jack's high-rise living room, Jack gives the team an overview of origins of life research, recent progress in the field, including efforts at his new lab at Chicago, and his optimism for what comes next. Beyond origins of life, they discuss Jack's unique path through science, including his work on CellX, an evolution-inspired process for selecting functional biomolecules from a random library, and his work on the scientific advisory board at Moderna. Jack also tells his story as a young man searching for interesting problems off the beaten path in the early days of molecular biology, eventually leading to his Nobel Prize-winning work on telomeres, the sacrificial repeat sequences at the end of DNA that govern the number of times a cell is allowed to replicate.
Cool. What initially sparked your interest in genomics?
My independent career started off, I was actually studying recombination, which is a project that developed out of my work as a postdoc. So we're trying to understand how DNA molecules repair damage, especially double-strand breaks. So that's something I worked on for, 10 years or so. And then the whole telomere project got started almost by accident, just because I met Liz Blackburn at a meeting. We had a great conversation, and I thought there were a few things that we could do to clarify the significance, and so that developed into also an important project. I didn't work on it for all that long, maybe 7 years altogether, before I felt like so many people were coming into the field that nothing we did would really make that much of a difference anymore. So that's when I started branching off and doing different things.
And originally, what got you into science? Like, how did you decide to become a scientist?
Oh, I was interested in science from when I was a little kid, interested in math, physics, chemistry. I had a basement chemistry lab where some dangerous things happened. And then when I started college at McGill, I got interested in biology and moved back and forth between biology and chemistry for quite a bit.
What was the kind of, like, if today, I don't know, if you're a computer scientist, the hot thing is like large language models.
Right,
what was the hot thing in biology when you were first getting into it? What was the siren call of like, oh, you got to work on this problem because everybody's excited about it?
Well, that's the thing. There's so many problems in biology, right? I got interested in nucleic acids and DNA. So damage to DNA is like a huge thing, right? It's a big problem for us, for medicine, for cancer. So understanding how DNA gets damaged and how damage can be repaired is an important aspect of biology. But that's only a small fraction of the whole space of problems in biology.
At what stage of your career was that? Was this already as an undergraduate or later on?
As an undergrad, I worked on more obscure problems. I worked on a green alga called deuterina. It's actually incredibly beautiful. So it's actually my first paper is on this green alga and how it makes a hormone for quorum sensing. But then when I went to grad school, went through some different projects that finally ended up, this was the early days of molecular biology, right? Cloning and gene manipulation. So I sort of got into that.
So this was before the era of next generation sequencing and buying kits online or whatever from suppliers that just do stuff for you magically. What was it like doing research in molecular biology back then?
You had to make everything yourself. One of the lab jobs that rotated was to make different restriction enzyme preps, to make different radioactively labeled preps of ATP, for example. Yeah, I mean, it was, compared to what we have today, it was very primitive. The lab that I was in was actually on the forefront of developing technology for sequencing. I think in retrospect, they didn't have enough chemistry to actually win out in the end, so they kind of got left in the dust. But we were on the cutting edge of a lot of developing things that are standard tools now in molecular biology. So it was an exciting place to be.
I'm also looking at the history of studying the broken DNAs.
Yeah.
And I was looking at that back in, it started quite early, like back in the 30s, or I don't know, even before that.
Yeah,
There
sure.
was Mueller and McClintock, who noticed that the chromosomes, they don't stitch to each other. And then the telomeres were already introduced at that time.
Yeah, so the idea is like in normal chromosomes, right? The ends just stay as ends, they're stable. But if you break a chromosome in the middle, those ends are very reactive, right? They'll either stick back together or they'll start doing all kinds of crazy rearrangements that are usually very bad.
And that's on what time scale does that happen? So it's not on like a chemistry time scale. It's more like a days, or like if you cut a chromosome, on what time scale does it start to?
There's a range of time scales depending on what happens. So just sticking back together is enzyme-driven cascade of reactions. So that can be pretty quick. But once you make certain kinds of rearrangements, then there's a cascade of events that can go on over many cell generations.
And those, I forgot the name, was it
Tetrahymena.
So what was special about it? So I read about that it had two nuclei. So one contained the broken piece of DNAs, and the other one is like the whole one. Why did it have
one
that?
is like a normal nucleus, the micronucleus, and then it also makes something called the macronucleus, where the long chromosomes are broken up into smaller pieces, and they all get telomeres attached to them so they can replicate as stable linear molecules. And so this is what Liz Blackburn was working on. And in particular, the ribosomal DNA is a very high copy fragment of DNA. And so you can get enough of that DNA and enough of the telomere sequences to work with it without cloning, right? Because when Liz started working on this, we didn't have clones of this. We didn't know how to do it. The technology was a lot more primitive than what we have now. So she was able to actually sequence telomeres from tetrahymena without having to clone them.
This is pre-PCR?
Yes, a few years before PCR.
In order to work with any DNA sequences at all, you needed to purify them in sufficient quantity. And if they weren't being made in large quantity, what can you do?
So then you could clone pieces of DNA. So cloning technology had been developed by that.
Meaning like putting it in yeast and having it grow more of it, or what?
Well, but the problem was for a piece of DNA from the middle of a chromosome, right? You can cut it out with restriction enzymes and clone it into a vector, a plasmid, or whatever. But if you want the ends of a chromosome, right, then you can't really do that because the ends are,
So you could. The problem was like actually getting the ends. Like it was almost impossible to read them because they would disappear in the process.
You could purify those fragments out of tetrahymena because it's abundant enough. But from other organisms, you couldn't really do that, right? But part of the problem was tetrahymena is so weird, right? It's got the whole thing with having two nuclei. Its cell biology is so strange that even though Liz's results were like really fantastic and interesting, people were wondering, what does this have to do with normal organisms or normal biology? So what we were able to do was take that DNA that she had isolated from tetrahymena and put it onto a yeast vector so that it was linear. So we had the tetrahymena ends on both sides. And then we said, well, we'll put it into yeast and see what happens. It was a long shot experiment. We didn't expect it to work, but it did.
So how long did it take you to do that experiment?
Well, the longest part was that I got the DNA from Liz, put it in the freezer because I was busy doing other things, and it sat there for 6 months until I got it out, did the experiment, and yeah.
Was this considered a very obscure system back then? Was Liz one of the few people who looked into this?
Yeah, because there were very, very few people working on tetrahymena. Liz, if I recall correctly, did the original sequencing of that DNA in Fred Sanger's lab. She had started working on it even earlier with Joe Gall. Yeah, it's definitely not one of your standard organisms that people study.
Do you know what was the motivation? Like, why did she decide to look at those tetrahymena? What was the motivation? I mean, I understand, like, maybe like zoology studying different types of, I don't know, what belongs with algae, bacteria, or
Tetrahymena is a ciliate.
Ciliate, that's right.
Yeah. I think there should be more of that, right? I mean, biology is so broad and diverse. If you restrict yourself to studying the standard organisms, you miss a lot of interesting phenomena. So I think it was actually a brilliant move to go outside of the normal range.
The situation where if you put a linear piece of DNA into yeast, it kind of falls apart, gets destroyed, doesn't get cloned, was that a known thing, or that also was kind of...
Well, that's partly how I got involved in it, because when I started working on recombination, what we were doing with my friend Rod Rothstein was we would take linear pieces of DNA, put them into yeast, and see what happens. And if the ends are not derived from yeast DNA, they just get chewed back. Occasionally, they ligate into a circle, which could be propagated. Mostly, they just get destroyed. And so the idea of putting something on the ends that would stabilize them in a different way was appealing.
So there was almost like an engineering need. Like, we want to put linear DNA and clone it in yeast, but it doesn't work. Then you have these linear pieces in another organism that do stay stable.
Yeah. So we thought, well, let's see if it works. Then we'll be able to study them. Then maybe we can see what's going on at the ends of yeast chromosomes normally. And that's interesting because of this older work by Miller, McClintock, and others showing that something special about telomeres. But nobody knew what it was.
And it's interesting that when it was decoded, it's that genomic mantra, I guess, GGGGTT. Is there an explanation of what is this particular sequence? And is it only for this tetrahymena, or is it exactly the same ending in human cells and other bodies?
It's slightly different in human cells, GGGATT. So one base different in that repeat. It's pretty close.
Different mantra.
But in yeast is kind of weird because it's G's followed by a T, but different numbers of G's. So it's not a simple repeating sequence. So experimentally, what happened is we put these tetrahymena telomeres into yeast, right? And it's being propagated in the yeast cell. And then we get it back out to look at it. It had gotten longer, and it had added this different sequence. That was the clue that said, okay, there's an enzyme that makes that DNA, and that enzyme is called telomerase.
How, in the end, did it come out that there was an RNA? And is it just co-purifying the RNA and the protein, or that it was a complex of RNA and protein?
Telomerase is a ribonucleoprotein. So the protein part is basically a reverse transcriptase enzyme. Mm-hmm. The RNA part of it is a template that actually defines that sequence. That repeating sequence is coded in the RNA template. That's why different organisms have a different sequence in their telomerase RNA. And the rest of the RNA is like a scaffold that brings in different proteins that do different jobs.
And how long did you stay in the field? So you did the discovery of, what were the other contributions into that area before you left it and started to look for something else?
Yeah, I think the last 2 years, Vicky Lundblad was a postdoc in the lab. And she wanted to make mutations and see what the phenotype was in yeast. And it turned out to be actually super interesting. So if you have a mutant in one of the telomerase subunits in yeast, initially there's almost no phenotype. But as the cells divide through more and more generations, they get sicker and sicker, and eventually they die. And what's happening is their chromosomes are getting nibbled away at the ends, right? Because they don't have telomerase to add back more DNA. And so that's turned out to be a very
This is a mutant
thing.
where the telomerase is not functional.
Right.
Okay.
So it's a senescence phenotype.
Right.
It's a delayed phenotype. You have to go through a certain number of generations before the telomeres at the ends of the chromosomes get nibbled back.
Was there a moment where you or the people working in this field felt like, oh my God, we've discovered the key to aging or something like that? Was there like a euphoria moment where it seemed like, okay, just around the corner, we're going to figure out? Because I think since, right, it's turned out, okay, this is way more complicated and it's not just telomeres. Right. But was there like a moment where everybody was super euphoric and excited, like maybe we've unlocked?
Well, yeah, when we saw that senescence phenotype, that was pretty exciting. I would say it maybe wasn't totally a surprise because, once we knew that the job of telomerase was to add back DNA at the ends, because otherwise they always get shorter, we kind of expected that would be the result. So that was the connection to aging. And aging is super complicated, right? There's lots of other things involved, but it's part of it. And then the flip side of that is cancer. And so other people found that in tumors, 90% of them have elevated levels of telomerase, which partly explains why those cells can keep dividing and dividing without limit.
Mm-hmm. So it's like with the cancer, so what happens? So you have, let's say, a group of cells with too many telomerase, and then do you just try to kill them, or you try to mutate them in a way that they go like fewer cycles of telomeres?
So what happens, what we think happens during the development of a tumor is first you get a mutation or mutations that release the controls on division, right? So the cell starts dividing more than it should, or in the wrong place. And most of the time, those like nascent tumors will die out because they have very low levels of telomerase. Every now and then you turn on extra telomerase and now they can keep going and you can start accumulating more and more mutations and grow more and more.
Okay, so somehow they get an extra of it. Maybe also like for the aging or anti-aging side, so since the telomerase is in every single cell, so how would they, you just inject them and then just let it propagate to all the cells somehow magically?
And then you get cancer.
If only it was so simple, right? If only it was so simple. So telomerase is one of the most tightly regulated enzymes in biology. And so most of our cells have extremely low levels of telomerase for this reason of protecting against tumor development. And so your stem cells are basically only allowed to go through a certain number of divisions. And that gets us to, like my age, right? And then your stem cells are not working so well and tissue regeneration starts to not work so well. And so the idea early on was, okay, well, if we could just figure out a way to turn on telomerase, maybe it would fix those problems or at least delay them. But then you have the flip side of the coin. It's one of those classic trade-offs or yin-yang situations, and lots of people still studying it, trying to find if we could turn on telomerase at the right time and place for just the right amount, maybe that would be good. We'll see.
Do you personally follow any life extension type protocols of supplements, or I guess the regular things of exercise and diet? But is there anything in your research that has inspired you to bet more on one thing over another?
I think diet and exercise are the kind of obvious things that we know work. Personally, very interested in, there are some metabolites like NAD that are known to decline with age. So I actually do take an NAD supplement. It's better studied than a lot of other things, but it's still not totally clear how effective that will be for different aspects of aging, but it seems harmless.
Have there been any interesting studies where you actually see effects like at the animal level? I know that they use telomerase to like extend the lives of cell culture and so on, but like at the animal level, have they had things where, we gave these mice cancer, or we made them live longer, or whatever, like these kinds of things? Have there been any like notable results like that?
Yeah, there have been quite a lot of studies with mouse models, that some of which actually look pretty promising. But mouse biology and human biology are pretty different, actually. So it's not clear if those results would translate well to humans.
Has anyone done something like taking out a human organ or a piece of an organ, let's say a piece of a liver or something, and then trying to de-age that chunk? Because if you cause cancer, you just don't put it back, something like that. Like, has anyone tried organoid or organ type systems?
So I think there are people working on all kinds of aspects of this. It's not something I follow in detail anymore.
So what happened? So you have done this great discoveries in 80s, 90s?
In the 80s.
80s, right? And then, why did you decide to move to another area, and where did you go?
So there were a lot of factors. One factor is actually when I was an undergrad, I worked on mice for a summer. And it was horrible. And I said, I'm never working on mice again.
But they did let you work in the lab. That's quite unusual, at least.
Well, the Jackson labs had a summer program for undergrads. It was a fantastic experience, but I didn't want to work on mice after that. So, and the whole field of, if you want to study telomeres in a real animal model, that's what you have to do. That was one reason I wasn't interested in going in that direction. The other reason is there were so many people, right? There must be hundreds of labs now working on different aspects of telomere biology. And I've always preferred to work in areas that aren't so competitive. There's many interesting problems out there. You don't have to work on the same thing that everyone else is working on.
Do you think that's a good strategy to take in general? To not go into the area of LLMs right now and AI and then look at the paths less taken?
Yeah, I mean, that works for me. Different people are different, right? Some people like to be in the thick of things and they don't mind if there's a lot of harsh competition. For me, I'd rather be able to sit back and think about things and find some interesting project that not so many other people are working on.
Do you think that used to be easier given the funding landscape of the 80s compared to now? Because there's a lot of people complaining about the short funding cycles, but I don't know how it was in the 80s.
I think it was probably a little bit easier to get money for non-standard things back then. But on the other hand, people these days, I think they do appreciate the fact that there's a lot of interesting discoveries to be made in different parts of biology. The development of DNA sequencing technology has allowed people to go out and study all kinds of weird organisms, right? So I think that's a great thing. But maybe the problem is for a lot of science funding, there's an increasing demand that it be translational. There has to be some useful application. And I think you need to find the right balance for that.
And you need to work with mice for that, more likely.
Even the clinical studies later, at later stage. What was the next area you landed and tried to explore?
So I was consciously looking to change into a different field, right? And I thought about all kinds of different things. I thought about going into neuroscience, but the technology there was so primitive. I just felt there was nothing that I could do that would be interesting there. The thing that sort of caught my attention was how do new molecular structures evolve? And so that's when we started doing what's now known as CELEX, making huge libraries of different sequences of, initially it was RNA and then DNA and then proteins. And, it's just saying, can we use the methods of molecular biology to evolve new interesting sequences that do interesting things? So I worked on that, and it was... a lot of interesting things came out.
How does CELEX work?
So the basic idea is you make a huge library of random sequences. So when we started this, we were doing this with RNA. So you can make easily, say, 10 to the 15th, or even 10 to the 16th, different random sequences. And then you want to find in that pool of 10 to the 15th sequences very rare molecules that do what you want them to do, like maybe bind to a target or catalyze a reaction. So you have to have a process that enriches for molecules that do that. And then there are very few survivors, right? Because most molecules don't do that. So you want to filter them out. So you do an enrichment, but the right molecules are probably still too rare in there. So you have to then amplify them. This was all happening right around the time when PCR was being developed. So you can amplify the population of survivors and then repeat the selection, and then amplify, and then repeat the selection. So it's just like the process of evolution with bacteria or organisms, except we're doing it with molecules. You go through enough rounds of selection and enrichment, and eventually the population's taken over by molecules that, hopefully do what you want them to do. And then you can clone them and sequence them and characterize them and see how they work. And that's the process.
So what was those, let's say, metrics you were looking in those molecules, what you were trying to synthesize? Or was it more like a general approach?
I mean, it's a very general thing. We started off for the first few years just trying to find RNA molecules that could bind to small molecule targets, just like antibodies can bind to a target, right? So I had been working on RNA ribozymes and everything for 5 or 6 years by that point. So I guess we skipped over a few things. But so I've been working with RNA. We already knew from the self-splicing introns that RNAs could bind a substrate, a small molecule substrate. So I thought, well, maybe we could get RNAs that bind anything. And doing this selection enrichment technology showed that, yeah, you can. Being able to bind a target is not something that only proteins can. RNAs can do it. Later, we found DNAs can do it. So in my lab, this was all developed by Andy Ellington. And very similar approach was taken in Larry Gold's lab at the same time. Yeah, and it turned out to be a very broadly applicable technology.
And there's a peptide version of this, which I poorly understand but find fascinating, where somehow you are actually enriching the protein that the RNA codes for, or you're enriching it with the RNA attached to it so that you have like a label that tells you exactly what it is you're enriching. Because there's no way there's no PCR for proteins.
Exactly.
So you need the DNA or RNA label.
Yeah. You need the information coded in RNA or DNA. So that's getting translated to make the protein, right? But once you've made the protein, normally it floats off and it's not attached to the RNA. So there's no way of getting that information back easily. So we just developed a trick to make sure that the RNA would be attached to the protein that it coded for. Okay, so you make these chimeric molecules that are part
you make
RNA.
It sound easy, like a trick, but I wouldn't even, where do you even start to make that happen? Like how do you, because the standard process is you have the RNA go into the ribosome and it comes out and then the protein comes out. So like what do you do?
So there's an antibiotic called puromycin. And what puromycin is a molecular mimic of the end of tRNA. Okay. So it goes into the ribosome, it accepts the nascent peptide, and then that causes the termination of translation. The trick we developed was we make our library of messenger RNAs. We chemically put puromycin at the very end. And so this construct gets translated, goes into the ribosome, you're making the protein, and eventually the ribosome comes to the end and it sees this puromycin and it transfers the protein to the puromycin, and there you go. They're attached.
And so basically the bridge is the puromycin.
Yes.
Okay.
Yes.
Oh, that's really interesting.
And was this the first idea you had as a solution, or did you try other things that failed?
That was basically the approach, from the beginning. It took a while to play around with it and get it to work efficiently. Yeah.
In general, what would you say is the ratio of things you have tried that failed and things you've tried that worked? Are you very, very lucky or very good at planning your experiments?
We try a lot of things that don't work, right? I mean, that's pretty normal in science, right? You come up with ideas, only a fraction of them work, and then you discard the failures and go on and try something else.
Can you think of one that frustrated you the most, where you thought, this should be working, but it just
should be
is
working
not?
100%. What is the biggest disappointment?
You'll have to let me think about that. I tend to block those things out of my memory, I think.
That's a healthy response.
Yeah, right. You don't want to focus on the failures, right? You got to move on.
So it seems like you have been almost all your life working to some degree with RNAs. Maybe RNAs are one of the key elements of
either DNA or RNAs. Yeah.
So when did you start thinking about asking the bigger question, let's say, where the life comes from?
Right. So as I said, during the 90s, we were doing this sort of CLEX technology, first isolating RNA aptamers that combine targets, then using that technology to isolate RNA enzymes, ribozymes, that can catalyze different reactions. And then we spent a while doing similar things with DNA, which surprisingly can also make folded structures and bind targets and catalyze reactions. And then the protein application, right? Making peptides or proteins that can do things. Doing evolution with populations of molecules in the lab is easy in the sense that we have all the fantastic technology, all the tools, the fantastic grad students who can do all these experiments. But somehow, this all got started on the early Earth all by itself, right? And so I started to get more and more interested and obsessed with that problem. So the last 25 years, that's been focus of the lab.
In terms of origins of life, what question do you think is more interesting? Whether exactly how it originated or whether it can be done again, like in a test tube? Like which of those two? Because I think those are two kind of competing questions.
Well, they're related. I don't think they're competing. I think, if we could build simple forms of life from scratch using any technology that we have available in any chemistry and still get something that could grow and divide and evolve, that would be super interesting by itself, right? It might not be related to how life got started on the early Earth, but I'm sure it would give us insight into how life got started all by itself. And so some of what we do is just directed towards getting systems that work in terms of, growth or division or genome replication or evolution, using maybe a chemistry that we have available in the lab that might not be the same as what was available on the early Earth. But it gives us a model system that we can work with, that we can learn from. And as we go through iterations of that, we can get closer and closer to something that could have actually happened spontaneously on the early Earth.
And it might be equally interesting, if not more, to have a different pathway or different types of life generated in the test tube.
Yeah, that's the other direction this can go. I think trying to design new forms of simple life where the chemistry is completely different or even a little bit different, it's also a super interesting problem. So we've worked a little bit with things that are fairly close to the nucleic acids we see in biology, just slight modifications. And so that's been interesting and informative. We've spent a little bit of time working on like a really different chemistry. Could you make a genetic material that could replicate in an organic solvent? So not in water, but in something non-polar. And that's like a super challenging problem, right? Really hard. We have a few papers on it, but we only got to being able to copy something that's 3 units long and with a binary code. So it's a long way from what RNA and DNA can do. I think that's a big challenge for chemistry.
And then maybe we end up with something that is silicon-based and not carbon-based life forms, like from the same period. Although I think that would be difficult with metabolism.
I think that would be really, really difficult. The chemistry of silicon, it's interesting, but it's so restricted compared to what carbon can do.
It's have the same extension of this question. So why there is only specific chemical elements are used? Because there was like other, let's say, like iron, or one of the most abundant on Earth is silicon, or even like sulfur. You would mention in this book I was reading about volcanoes. Yeah. So it seems like a lot of that stuff happened in those conditions, but somehow we don't have no sulfur, iron, or silicon in the RNAs, DNAs.
So you don't find in modern RNA iron, for example, but iron is very important to our biochemistry. And iron, I think, was even more important to the early chemistry that gave rise to the building blocks of RNA, the nucleotides. And for sulfur, there's not very much sulfur in modern RNA, but there actually is sulfur in many tRNAs that are part of the translation apparatus. And there's very good reasons for thinking that sulfur played a more important role, maybe in an earlier version of RNA. There are nucleotides that actually have sulfur atoms in them that could be precursors to the modern nucleotides. This is all work from John Sutherland's lab.
But it seems like also from reading, the holy grail of the nucleotides is the cyanides. As I understand, so it's like a CN bonds and the rings. Do we have any intuition why those CN rings are in the heart of the building blocks for life?
So cyanide itself looks like a fantastic starting material. And in the Sutherland lab, they've worked out very efficient and beautiful pathways to getting to the nucleotides. Why those are the only molecules? I mean, that's the question, right? We don't know if that's the only way that things could work, but it's a way that could have happened, that the chemistry of cyanide can get you to nucleotides, which can get you to polymers that code for information in a heritable way. Are there totally different ways that life could get started? We don't know. We just know that that is one way it could have happened, because it did happen.
Because it seems somewhat paradoxical given how toxic it is to us.
So cyanide is toxic to us because we breathe oxygen, right? And it complexes with hemoglobin and messes up that process. But cyanide early on is a great starting material because it has carbon and nitrogen, which are abundant in RNA and DNA. And it has that triple bond that stores a lot of energy. And so you can do reactions with it that go forward because you're using that energy to build more complex molecules.
While reading, it seems like there was a big revolution in understanding how those protocells were formed. So let's say for nucleotides, there was, let's say, like, I believe 3 components: the base, the phosphates, and the sugars.
Right.
And then you describe in the book that actually it was maybe not about like how to assemble those pieces together, but what were like the chemical reactions which evolved into bringing those substances.
Yeah. So if you look at the structure of a nucleotide, right, it's just natural to divide it into the nucleobase. So we have UCG in RNA, and that's what codes for it's the informational part of it. And then those are attached to a sugar, which is attached to a phosphate. So it's just natural to divide it up in that way. If you try to take those pieces and put them together, it's very hard to do. You can do it with enzymes. Biology can do it with enzymes. But it looks like there's a much easier and more efficient way to do it chemically, that it goes through a different pathway. It's counterintuitive, which is why it took a while to get figured out.
So it was surprising at that time that you could do this without enzymes?
Well, that's the whole problem with prebiotic chemistry, right? Is by definition, there's no evolved machinery. So the origin of life, there's no enzymes, there's no ribozymes, there's just chemistry and physics, and that's what you have to work with.
What's
so it's a big constraint?
Yeah. What's kind of if you think about like just on a very physical level, like what today can we put into a tube and get an interesting result? You know, not so theoretically, but just physically, like if we start mixing things, what's like the state of the art from that perspective?
Okay, so this is also an area where there's been a change in thinking. A little over 70 years ago, here at UChicago, Stanley Miller did the famous experiment, Miller-Urey experiment, right? Putting energy from a spark discharge through an atmosphere that at the time they thought was what the early atmosphere would be like. So methane, hydrogen, ammonia, stuff like that. And you got amino acids or amino acid precursors. So it was revolutionary at the time, right? But if you look at the outcome of that kind of experiment, what you get are thousands, probably millions of compounds, right? And the ones that you actually want to build biology are a very, very, very small fraction of what gets made. So it's actually not a good way to get started. What the change in thinking has been to try to find pathways that give you a lot of just a few of the right thing. And to me, that's epitomized by the work that John Sutherland's been doing and his colleagues and students. And they've found very beautiful pathways that give you high yields of a few products which are on the path to biology.
So in the Miller-Urey experiment, actually, the amino acids they did find, they were all racemic instead of chiral, which is what we observe. Is this similar for this more restricted pathway type work? Do you still get racemic mixtures, or do we have clues to why there's homochirality?
So the problem of homochirality is fascinating. It has a long history. People have been searching for mechanisms that would have given rise to homochiral building blocks of biology, like for a long time, right? So there are a lot of mechanisms that have been proposed, most of which don't really work very well if you dig into it. And if you think about what you really want, you don't want just something that works on just one compound or maybe just on, say, amino acids. It turns out that in order to replicate RNA without enzymes, you have to have homochiral nucleotides. And that means you need all 4 nucleotides to be homochiral. So the best way to do that would be to have a homochiral precursor that would go on and make all 4 homochiral nucleotides, right?
But then you're shifting the question, where is this homochiral precursor?
It's very important to know where it came into the pathway. Right? So there's been a very exciting result that has come out of work that's been done by Furkan Öztürk when he was a graduate student with Dimitar Sasalov at Harvard. And what they focused on was a molecule called RAO that is a precursor of the nucleotides. And it's been an interesting molecule for a long time because it tends to crystallize out very beautifully. And it crystallizes in a special way where it makes separate crystals of right and left-handed versions of itself. So now the problem gets reduced to, can we somehow get just the crystals of the chirality that we want? And so it turns out what Furkan was able to do is there's an effect called the cis effect, chiral-induced spin selectivity. There's a process that brings together spin selectivity of electrons in molecules. And so in a magnetic field, turns out you can actually get this RNA molecule to crystallize for one-handedness, to crystallize preferentially on a magnetic surface that's biased by a magnetic field, which could be the Earth's magnetic field. And so this is something that actually works in the lab. They're figuring out how well this could work in nature is a current challenge. But I think it's by far the best explanation that's come out so far, in part because it brings in chirality at the right place in the chemistry. So it's an exciting advance. There's a lot more to do, but it's a potential answer.
It's certainly a lot more efficient process than what Louis Pasteur had to go through by tediously sorting his tartaric acid crystals in with the tweezer.
Yeah, that's something that couldn't happen in nature, right? I mean, there are things that can happen in nature. There are ways to, if you have a small bias initially in favor of one-handedness, there are physical processes, including crystallization, and that can amplify that bias. It may be that this enantioselective nucleation of crystallization might have given an initial bias that was then amplified by some of these other processes. That's the kind of thing people are thinking about now.
And what is the current state of the art for developing protocells? I have heard about that you can create, say, some fatty acid compartments where you put some chemistry inside.
Yeah.
Are there any certain conclusions we can make right now about how the life started, or what's the projection in this research?
So this is basically what we've been working on for the last 20 years. I would divide that overall problem of how to make something that looks like a protocell. I would divide it into 3 areas. So one area is how to replicate the genetic material, say something like RNA, maybe a primitive version of RNA. We have to be able to go through cycles of replication, right? Just using chemistry and physics, no enzymes. So we've made a lot of progress on that, but we're in the middle of a lot of exciting advances. The second big area is the cell membrane. You need some kind of compartmentalization. You can't just have all these molecules floating around in solution. In all of biology, cells have membrane boundaries. So it seems logical that the first cells must have had some membrane boundary. But then the question is, well, what would that have been made of? Modern cells have very complicated lipids in their membranes. Primitive cells probably had much simpler membranes. The ultimate in simplicity there is just fatty acids. So we've studied membranes made of fatty acids for a long time. It's not clear if that's sufficient or if something of more complicated mixtures might be required. That's one of the things we're trying to figure out. But those membranes have to have the right property. They have to form spontaneously. They have to be stable enough. They have to let small molecules across. Nutrients that were made in the environment have to be able to get across the membrane to the inside, where they can take part in things like RNA replication. And then the third area is getting everything to work together. So the RNA chemistry has to be compatible with the membrane chemistry. And that's also a big problem. We have artificial ways of getting around that, but there has to be some way that would work in nature that everything could be compatible. And so that's a whole different series of problems that have to be addressed.
What do you think of like the, I mean, I'm not too familiar with this, but I know that there's an idea that there might have been like mineral membranes where basically you have a very porous rock formation or something and things can grow in the pores and have somewhat of a membrane type behavior where you can have diffusion through the pores between the compartments.
Yeah. At this stage, we know so little. It's worth exploring all kinds of things, all kinds of alternatives like that. I'm not a big fan of the idea that the actual membrane could be a mineral composition. They're very porous rocks. A lot of volcanic rocks are very porous, and so they have little micro compartments in them. So that's an interesting area. Dieter Braun at the LMU in Munich has been studying this. It's very, very interesting physics of fluids in these complicated porous rock environments. But at some point, primitive cells had to have a membrane boundary. That's just the part of it. There are other possibilities that we and other people are also studying. There are these complexes called coacervates. Right? So if you take polymers that are positively charged and negatively charged, right, they'll spontaneously come together in water and they'll make sort of liquid droplets. Those are interesting chemical environments. We're trying to see, are there things you could do in those liquid environments that would be helpful? And maybe they could form inside membrane-bound vesicles. There's a whole range of different phenomena that are potentially relevant and really need to be studied.
If you take an engineering approach and say, let's take some pre-made components, for example, RNA or whatever it might be, right, hold that constant and let's make our own membrane, for example. Have researchers had any luck with doing something like that, where reusing some existing components, but then building a new one, for example, building a new membrane and putting it around like cell-free media or whatever, kind of like Frankensteining together a cell?
Yeah.
Does that look promising to you, or has anyone had any luck with that?
Well, there is this whole field of synthetic biology, right? What a lot of people are doing, this is actually probably a bigger field than origin of life per se, but people are trying to take apart existing cells to components like the membrane, the cytoplasm, the DNA, and see if you can put them back together. So it's also very interesting and probably very informative about how cells work. It's not directly related to how the first cells formed. So when we're trying to mimic more of early steps in the origin of life, we do things like we make membrane vesicles out of, for example, fatty acids. We encapsulate small bits of RNA in there. And we try to do replication chemistry inside these membrane vesicles. So for us, that's the closest that we are getting, right, to trying to actually have a protocell that can really replicate its genetic information and also grow and divide. We're like actively trying to do that and make it work better as we learn more.
So how much of the evolution do you see on the self-replicating RNAs inside the fat acid compartments?
So for us, the holy grail is to be able to watch evolution start to happen. Right? We're still a ways away from being able to do that. The first thing we have to do to get to that is to have a system where the RNA can actually replicate inside vesicles. We have ideas about how to do that. We just have to make it work.
So it's like the chemical compatibility of the vesicle with the RNA synthesis.
Yeah, we have artificial ways of making things compatible. So we want to find ways that would be more natural. We can do experiments where the membrane system is compatible with the RNA chemistry. So we do experiments like that.
How big are the RNA pieces that are replicating in this kind of models?
That's the problem, is we can do copying, but we can't yet do replication.
How do you define those two? Like, what's the difference?
Yeah, so copying means, say we have one strand of RNA, and we want to build a complementary strand. So we can do that. We're getting better and better at doing that chemically. But then once you've copied a strand, you want to copy the copies, right? And copy those copies and just have this exponential process of replication. And so that's one of the big challenges.
Where's the bottleneck there? Like, so if you have 2 pieces of RNA that are bound to each other, or
yeah. So if you have 2 pieces of RNA that are base paired to each other, right? You can get them apart by, for example, heating things up. But then when you want to copy those strands, you have to change the conditions so that base pairing is possible again. So you cool them down, they immediately come back together again, and now you can't copy them anymore. So figuring out how to get around that problem.
So the real difficulty is that once you've made 2, how do you separate them permanently, like long enough to make
long enough to do the very slow copying chemistry. So during the pandemic, there was a period we couldn't even go into the lab. So there was nothing to do but sit at home and think. So we came up with a model that is called the virtual circular genome model. And I think it's a way of getting around that problem. We're studying it, but we'll have to see. So the idea is, in bacteria, typically you'll have a circular genome sequence, right? And so in a very early cell, you could also imagine having a circular sequence, but much smaller. The thing that's different about the virtual circular genome model is that the circle is virtual. There are no actual molecules that are circular, just fragments that map to the circle.
They connect. They have some overlaps, and they connect.
Yeah, you could start and stop at all kinds of random places, and you could have both strands of the double-stranded circle, right? So all those pieces can come apart, come together transiently, do a little bit of copying, come apart, come back together in a different configuration, do a little more copying. So instead of doing what happens in biology, where you start copying at one place and you go all the way around to the end, here we're doing little bits of copying all the way around the circle all the time. And so you feed in new small pieces, they grow longer, eventually that process gives you replication. Is that the way life actually got started? Can we actually make it work? We don't know yet, but this is one of the things we're exploring in the lab.
Maybe like some simple question for those. I'm an engineer, I'm not in biology. It's just like in general, the moment of, let's say, division or like some sort of, like, let's say, dividing the cell. So how does the cell knows that it's time to divide? Like, is there some clock? Like, what triggers it? Or, for example, if it's also self-replicating RNA, why would it want to? It's like, all right, I want to self-replicate.
Right,
Let's do it.
So this is another big open question, right? How do you kind of keep things in sync? So we have all these ideas about how the RNA could be replicating. We have experimental results that we can take our membrane vesicles, we can feed them with more lipid molecules, they get bigger, we can see spontaneous division happen. We have different ways that can happen. So we have both sides of it, right? But now if you put everything together, how do you avoid having, say, the RNA replication keep going and going and going and then just burst the cell because it's full of too much RNA? Or how would you avoid having the membrane grow and divide and grow and divide, even though the RNA is not keeping up, right? And the RNA will get diluted out to nothing. So how to keep it synchronized is a big question. Again, we have some ideas, but yeah, that's one of the big unknowns.
But for normal cell, it's just everything is controlled by enzymes. Or?
There's incredible machinery, like alien machinery that controls it. Like, absolutely impossible to imagine how it evolved from nothing. I'm kidding, but it's an extreme. Have you ever seen the micrographs of like how the genes are pulled apart and all this? I mean, there's incredible machinery that divides the cell.
And none of that machinery.
Like a cytoskeleton and
Right. So that machinery wasn't there at the beginning, right? So probably keeping these two processes working together on the same time scale was not very efficient. But that would mean that there's a very strong selective pressure to evolve machinery that would keep things from getting out of hand in one direction or the other. We think there are some simple physical processes that could have helped at the beginning, and maybe evolution would build on those processes to keep the RNA replication in sync with vesicle growth and division. But there's a lot of interesting questions there.
Yeah, I think one thing, you might have mentioned this in one of your talks, or maybe it's from somewhere else, but it's like if you have the protocell in some kind of pool of water, and there occasionally are hot bursts of hot water that will then like pull it out into a plume of hot water, and the RNA can decouple in that hot water, a little bit of how PCR works, actually, right?
Yeah,
Very
yeah,
interesting.
right.
And then it can cool again later. And so maybe there are some cycles that you can get, maybe dry, wet cycles also in some kind of like physical environment. One interesting question that we haven't covered is just generally the landscape, like before you approach like the details of origins of life, what is the big picture landscape of like where did it originate? What kind of like, I'm sure you thought a lot about this, especially choosing like exactly what to focus on. What's your thinking on this? Just like the big picture questions of in what environment did it originate, kind of what chemistry was available, these kinds of things.
That's another area where the general thinking in the field has evolved a lot. So I'd say in the early days, the chemists would be doing their prebiotic chemistry synthesis in the lab and only concerned about getting the reactions to work and finding a path. But now we've gotten to the point where we can really think about, well, what kinds of environments in nature would allow the chemistry to work? And there's a kind of a feedback between the chemistry and the geology. The chemists say, like, I want this reaction to work. Here's the conditions that I've got it to work in. Talk to the geologists. Is there an environment where this could happen? And they might say, yeah, it could happen in a volcanic region, maybe in an alkaline carbonate lake, or someplace where you have wet-dry cycles. Or maybe they'll say, well, that's just not realistic. You have to go back to the drawing board and find some chemistry that can work in environments that we know would have been around. So this kind of feedback, I think, is very, very helpful. And it lets us focus on environments like impact crater environments or volcanic regions. So I'll just give one simple example. So we were talking before about cyanide being a great starting material for the chemistry, right? But cyanide's formed in the atmosphere and rains out as a very dilute solution, which will then just go and hydrolyze and not give you anything interesting. So how do you capture the cyanide and build up a reservoir? Chemically, that's very simple. If you have iron around, you can form iron cyanide complexes like ferrocyanide, and those could potentially accumulate and build up a reservoir. So that was a great theory, but what kind of actual natural environment could that take place in? So that led David Catling in Seattle to do some really interesting modeling of these alkaline carbonate lakes, which is a natural environment they've been studying. And they found that, indeed, under these conditions, which can happen in nature, ferrocyanide molecules could accumulate and could be something that builds up a reservoir. It's a great example of this kind of feedback between chemistry and people who study the geology and the geochemistry of natural environments.
So then what kind of environment is needed? Is like volcanoes, something carbon, nitrogen atmosphere, high pressures, gradients in temperature, UV. What is this list of, let's say, if you would like to have build a, I don't know, some sort of greenhouse, like in the lab, what do we need to do?
Yeah, so I think one thing that's pretty clear from the chemistry is a lot of the important reactions are photochemical. So you need UV light from the sun. So that means you have to be on the surface. So that kind of eliminates deep sea hydrothermal vents.
So that would be a big thing, right? So hydrothermal vents were this kind of like origin of life story for a while. Your feeling is that because of the UV requirement, it can't happen.
There are multiple reasons why the deep sea vents are not really relevant. It's hard to accumulate and concentrate compounds at scale in that kind of environment. Mm-hmm. If you're in an oceanic environment, everything gets diluted, right? And then nothing interesting can happen. People have studied the chemistry of those environments, and in my opinion, basically no interesting chemistry that gets you close to the building blocks of biology has been found. We were talking before about the crystallization of RAO. That can only happen on a surface environment where things are getting concentrated, you have evaporation, you have solid surfaces. Yeah, lots of reasons for liking surface environments. We need UV light. We were also talking before about sulfur chemistry. Where does sulfur come from? Volcanic outgassing gives you sulfur dioxide, which turns out to be really important for a lot of the chemistry, but also hydrogen sulfide. So if you've ever been to any place near a modern volcano or even like Yellowstone, You can smell hydrogen sulfide, right, from miles away.
It's like mineral water, right? It contains a lot of it.
Some of them contain a lot, right? So by studying natural environments, we can focus in on what gives you the right starting materials, what gives you the right physical processes so things can get concentrated, and what gives you a kind of the fluctuations in the environment that might be required. As you said, wet-dry cycles, light-dark cycles, temperature cycles, all kinds of things like that.
One of the things that I think probably the craziest like environment idea that I read was ocean spray. So having like droplets of ocean spray where the air is the outside of the membrane and the inside is just water. I don't know. What's your take on the ocean spray?
Yeah, again, there's interesting chemistry that can happen on those interfaces, which is definitely something that should be studied more. But I find it hard to imagine how you could have growth and division or how you could feed nutrients into... So I think the idea of membrane-bound protocells in a liquid environment makes it's easier to think about for sure.
Yeah, that's a good point. There's no mechanism for gaining anything from the environment.
I think these aerosols that are generated from waves and spray could be really important for dispersing early life around the planet. That's one possibility.
Is it also possible maybe to do some live space travel, send it to other planets, use maybe light sail?
Oh.
I don't know. How is it? I see like the biggest problem is probably the gamma rays, but maybe there is something else. Or could it be that someone send a similar light sail to Earth?
I mean, Avi Loeb claims this is what happened.
So I think when the solar system was young, especially when it was very young, there were a lot of large impacts, not just the Earth, but Mars and other planets, right? So there's a lot of transfer of material, rocks that got ejected after a large impact and eventually land on another planet. I think the transfer of life between planets within our solar systems is possible. Maybe we'll find life in some, like on subsurface Mars or in Enceladus. If we do, I bet it will be related to our life. It's a totally different ballgame when you're talking about transfer between planets and different stars.
Just because of it falls off with R squared, basically. The signal should be very small.
It should be a small signal, the radiation damage during millions of years in space. On the other hand, just in the last few years, astronomers have detected things from other...
Asteroids
that actually came presumably from other solar systems, right? That have come whizzing through our solar system and then just gone off again into space. So yeah, probably some fraction of those hit the early Earth, but what's the chance that they actually carried anything alive in the first place and then anything survived the impact and survived the journey? I'm personally pretty skeptical that could actually happen, but who knows?
So you recently published a book, Is Earth Exceptional?
Yes.
What would be your one-sentence answer to that question?
Well, my one-sentence answer is that's an interesting question, right? and we don't know. It's just so exciting that like half of astronomy these days is focused on finding exoplanets, trying to look at them in more and more detail, ultimately trying to get some signal that there might be signs of life on other planets. And that's really what we need to do, right, in order to know whether Earth is unique, incredibly rare, or we know that there must be millions of Earth-like planets orbiting sun-like stars just in our galaxy, let alone the whole universe, right? I think we all want to know, is there life out there, or is Earth somehow so special or so lucky that life only got started here? That's the big question.
What would you do if you were given a large sum of financial resources, or let's say even unlimited resources, what we have here, as many people work on that project? What would you do?
Certainly, we could use more money for the sort of small-scale lab experiment. We were very fortunate to have funding from the Simons Foundation over about a 10-year period. And I think that funding really revolutionized the field. It let people do all kinds of exciting new things. And it brought together this confluence of early geology and studies of planet formation in early environments with the laboratory-based chemistry. We need to continue that. We're very lucky to have support from some foundations now, like the Sloan and Moore foundations, but it's a smaller scale. I mean, there's so many fantastic young people out there starting their own labs, studying these problems. We need more support for them. On the astronomy side, the budgets are 1,000 times bigger.
Astronomical.
Right? They're astronomical, right. I mean, we need millions of dollars, but to do a space telescope, you need billions of dollars. So it's a whole different level of challenge, and the timescales are longer. I would find it very hard to be involved in a space mission, right? That it might take your entire life, your whole career, to see something come to fruition. Like, people worked on TESS, for example, for a long, long time before the harvest of the number of exoplanets came through. It's such a huge investment on long time scale. For us, we do experiments where you think of something, give it a try, and we know the answer maybe weeks or months.
Is there a technology missing? Like, PCR was like the key to a lot of things that came afterwards in biotech. Is there like a technology, like for example, origin of life experiment on a chip, that would allow much faster progress, rather than doing the experiments in like traditional format, doing them in some kind of, like CellX style, like some kind of mass parallelism, or, some kind of spectroscopy modeling? I don't know, I'm just making stuff up. But the question is, have you thought, like, what would be like a technology that could take the same people and same resources in a way, but then amplify them the way that some technologies have done for biotech and other fields?
Okay, so I would say two things. So first of all, we are always trying to use the best technology that's available. Mm-hmm. So modern DNA sequencing, for example, that is something that has now become integral to the experiments that we're doing. We can look at RNA copying chemistry experiments on a huge scale. What we used to do is we would take one RNA and see how well does our copying work, right? Now we can take basically all possible template sequences and do the same experiments and see how different sequences get copied, where the errors come in, what context the errors come in. So this new technology is extremely helpful. We're always trying to use the latest advances in microscopy, for example, to look at our protocells, understand how they're growing, how they're dividing. We definitely try to use the best technology that's available. On the other hand, this idea of parallelizing things always comes up. What if you could just try millions of things, right? And see. My answer to that is that what's limiting is actually the ideas, the concepts. And you can't parallelize that, right? You just have to have people thinking about the problem. I think that's where our real roadblocks have always been.
Perhaps maybe LLMs or AI can contribute to generating those ideas which would go into the labs.
Well, maybe. We'll see.
There's one skeptic here, at least, of that. I don't know.
But it's such an interesting answer, because that is literally also what Sirac and Soller, whom we have recently interviewed, who made the first design of a quantum computer, they said the same thing. It's idea limited.
It's the ideas.
Yes.
Yeah. I definitely feel like that. And the reason is, if we look back at the advances in the field of origins of life, this is something I tried to emphasize in my book, that the revolutions come from giving up on some fixed idea that everyone holds and realizing that actually things could work in a different way. And that's happened again and again and again. It's the advances in how you think about the problem that really lead to new development.
So how would you inspire younger minds to come to the field? Or maybe you have something else to tell them. What should they do?
Well, there are a lot of important questions in the field. So, we really need young people to come into the field. But I also have to caution them that it's a difficult field. You really have to be thinking out of the box. And it's also difficult for just funding and just being actually able to do this, because it's curiosity driven, right? We just are interested in it because it's such a fundamental question. How did we get here? But most funding agencies want to see something useful coming out of it. And
this is actually one thing I was going to ask about is like, if you, let's assume that with, again, we're getting better technology, people have been thinking about this for a long time, there's more clues. In 10 years, 30 years, whatever the number is, there's some kind of really meaningful breakthrough where it's like kind of solved. So we've either made life synthetically, maybe it's not the life that happened originally, or we figured out how it happened, whatever the case may be. What does that future look like? Like it's always hard to imagine this stuff, but I actually think that probably there would be a lot of applications. So I wonder what that future would look like. What would people be working on as a derivative of that? Basically, once you can make self-replicating nanotechnology, basically, like what does that mean?
So in terms of the evolution of life, I think where it's heading is that the next steps would be developing an understanding of, how did metabolism evolve? How did all this chemistry that used to go on out in more or less extreme conditions, volcanic regions and stuff like that, UV catalyzed, how did that turn into something that happens under very mild, gentle conditions inside cells? Thousands of reactions going on all the time in every cell, in every bacterial cell, every cell in our bodies. How did that develop? So that's something a few people were just starting to think about, some initial steps there. Also the process of translation, right? How do you have the genetic code giving us, proteins that can do all kinds of amazing things? How did that develop? That's something we've started to work on, little bits and pieces of that, but there's so much to do in understanding that. Membrane transport machinery, which is, fundamental to everything, like everything in neuroscience is about transporting ions across membranes, right? How did that all develop? I think once we solved the earlier questions of how did the first cells get going, then we can start to think more rationally about how they got more complex, how they started to adapt to their environment, explore different environments, and evolve all this internal machinery. So that's kind of where I see the field going.
Is your guess that once there's a self-replicating system, that things can go quickly in the sense that like it'll be possible to kind of speed up the evolution by doing artificial selection in the lab or things like that and get a lot of interesting results that way versus is there a good understanding of how long that took on Earth? No. Like was it a year, a million years, a billion years? Like is there like a rough...
There's an almost billion-year window of uncertainty there. So obviously the early Earth had to cool down to the point where you had liquid water. And so that's maybe 4.4 billion years ago, maybe 4.3 roughly. The earliest incontrovertible signs of life in terms of like microbial fossils are in the range of maybe 3.5 to 3.7 billion years ago. So there's a huge window there.
Are the fossils that we found already roughly like normal bacteria?
Yeah, these are basically stromatolite fossils, so they're probably similar to stromatolites you see
around today. Yeah. So we're missing everything
in between
right before that.
Yes.
Wow.
And we have billions of years to figure this out. Regarding the
maybe you do. I only have a few years left to figure this out.
Well, we fix the aging problems now with telomeres. But regarding applications, let's say from genomics, we know that there are some other barriers for gene engineering, which comes from policies or ethics-related things. Yeah. So do you see similar restrictions for applications which would come up from origins of life research?
So I think origins of life research, and even making these simple self-replicating systems, I don't see serious, ethical limitations or sort of hazards of doing this because it's so primitive and we have to actually supply everything that they need for replication, which is not happening in a way that is happening in biology. I think the situation is different when you start to look at synthetic biology and manipulating, for example, bacterial cells or other life forms. There are, I think, much more obvious hazards that we have to be very careful about.
You mentioned that you are trying to build the cell from scratch. Then there is, let's say, some other medical like treatments, which are not based like from the scratch type of solutions. Are there any other like gaps in between how like the bacteria or our body operate right now, which would still be helpful to kind of fill the gap? Like we don't have to trace back to the very beginning of how the life started, but are there still some gaps in between which would be interesting to find out the answers how something works, the complexity of the body?
Well, the use of RNA molecules in different kinds of therapies and vaccines has exploded over the last few years, right? So I got to see part of that up close because I was the chair of the SAB of Moderna from its beginning and then for about 10 years. So, the whole idea of making vaccines for messenger RNA is new, and Katie Carrico and Drew Weissman got the Nobel Prize for their early work on that. And one of the key interesting things is you actually need to use modified nucleotides to make it work. And so that raises the whole question of, well, there's so many RNA nucleotide modifications seen in biology today. Where did they come from? Did they come from early in evolution, or were they later inventions? So understanding all that, I think, is pretty interesting. More and more has been discovered about different roles that RNAs play in biology, right? So the most recent Nobel Prize in physiology or medicine is from the regulatory roles of microRNAs. So understanding the origins of that whole system, right? How far back does that go? Is that something that you could trace back maybe to the RNA world? Certainly riboswitches look like something that could have played a role in the RNA world before the evolution of coded protein synthesis. So understanding how a lot of this machinery that we see in modern biology that's RNA-based, how that all came to be, understanding it better potentially could lead to new ideas for applications.
And are there any, maybe some sort of like bounties in the science, like, okay, some things are wanted, and if those problems will be solved, maybe a Nobel Prize will be given. Are there any like list of challenges or, let's say, the directions which people try to go to do the research for those holy grail experiments? Or
yeah, microRNAs was one that just did win the Nobel Prize. I think RNA modifications is a hugely interesting area that could lead to a Nobel Prize. I'm sure RNA modifications are going to lead to a lot of practical applications as well. I think you can already see some examples of that. And understanding, the roles of RNAs in biological replication. We already know it's important in, for example, tumor development, normal biological development. So there may be all kinds of interesting developments in those areas that will come out in the near future.
So you just mentioned Moderna. How frequently are you involved with commercialization efforts in this space?
Yeah, so throughout my career, I've tried to keep the work going on in my lab focused on very basic questions. But translational research is also really exciting and important, and it's very satisfying to see something actually get developed to the point where it can actually help people. So I've been involved in a lot of biotech companies throughout my career, and I've found that to be very interesting and very satisfying. So some of the things that came out of the mRNA display technology that led to several companies and actually molecules that are in clinical trials. So seeing something go from something very fundamental question you're trying to look at in the lab and see it actually go all the way to something in the clinic it's a long path, but it's very satisfying. So I've enjoyed being involved in the more translational developments, mostly in companies.
If you had to pick one thing where you have a kind of like a view or an opinion or a position about science or related topics like that you think is a little bit heterodox, like people usually don't think about it this way, like the way you do science or something like that usually don't find that, like, it's not typical, what would that be?
I guess maybe when people look at my career, they see me working on things that are a little bit non-mainstream. So I enjoy that, as we were kind of talking about before, right? I don't like being in this super competitive environment. But the challenge is to find a question that's interesting that other people aren't already working on, but that is tractable. Right? There's lots of interesting questions that we don't have the technology. We have no idea where to even start. But finding something that's tractable and interesting, that's the way I like to find an area to work on.
Is there a more detailed list of criteria how you would look at the project or like think about it? And it's like, that one maybe not, that one maybe not, but this one, all right, I'm doing that one.
Yeah, so when I was trying to change fields back in the 80s, I decided not to go into neuroscience because the technology was too primitive at the time. Basically, the only way to map brain function to particular areas of the brain was from damage from trauma or stroke. Now, right, you have like higher and higher resolution fMRI methods and all kinds of other amazing optogenetic technologies. So that field's opened up. And if I was starting now, I might be tempted to go into something like that. Another direction I decided not to go in was just to focus on structural biology, because I couldn't see anything there that I could do that would be, something fantastic and new. There were so many other smart people developing new technologies. I didn't feel like there was a niche that I could contribute there. Yeah, then when I started to get more interested in the origins of life, just because of the question of how did evolution get started, that seemed like something where I could make a contribution, because after all, it's basically nucleic acids, chemistry, and molecular biology to a large extent. And not that many people working on it, and very interesting questions that are actually tractable.
Being a physicist, where do you think a physicist can contribute to origin of life research, apart from astrophysics?
Well, this whole question of the origins of chirality, I think, is a great example. It was really physicists who had been working on this cis effect, this chiral-induced spin selectivity. So that's a great example. But maybe more broadly, just sort of geophysics, thinking about how planets form, how protoplanetary disks, lead to the assembly of planets. There's a lot of interesting physics there, but also thinking about the environments on the early Earth. A lot of interesting physics to delve into there. And maybe more closely related to what we're doing, the behavior of these membranes. Membrane biophysics is itself a huge field. And so there's a lot of physics-related questions to how membranes behave in biology and in pre-biology. Also, I should say, one of the nicest things that's come about since I moved to the University of Chicago is I have a great collaboration with a guy in physics who's in the physics department, Arvind Murugan. And he and I first started talking about some of the problems of RNA replication and this virtual circular genome question. We wanted to be able to model that system to help guide our experiments, but it's pretty complicated because you have potentially thousands of components interacting and pairwise and tertiary interactions. You have sort of billions of possible interactions. How do you go about modeling something like that, these really complex systems? So being able to talk to a physicist about this and get them involved in this research has been really fantastic. So you get a different approach to the problems from someone with a physics background.
One question that just comes up, because you mentioned Moderna, is the lipid nanoparticle technology useful for origins of life research? Like basically the mRNA vaccines are little vesicles, right? So can the same equipment that makes, now that's been scaled up enormously, right? So we can make, I don't know how many, it must be trillions of lipid nanoparticles pretty easily. Sure. So is that useful for producing like protocell prototypes or whatever it might be?
It turns out the way that you make the mRNA vaccine nanoparticles is a little bit different. They're not simple membrane shells with stuff on the inside. They have a more complicated internal structure. But I think, coming from a field of having thought about making particles that contain both lipids and RNA was helpful in thinking about it, suggesting experiments that people could do to optimize things.
Since we also have quite developed semiconductor chip technology, this is more a curiosity-driven question.
Yeah.
Did you think of combining organics and non-organics? Like, let's say, RNAs with something like transistors or other nano-electronic components?
making a laser from RNA.
Cyborgs.
Well, you know, there is a kind of confluence of things like that in nanopore sequencing, right? Which is a very interesting technology that's being very rapidly developed. And also people use zero-mode waveguides to look at molecules at a single molecule level and relatively large scale. So I think technologies like that can help us to look at individual particles, which I think is very important. You know, if you look at things in bulk, right, you just see the average, right? But what you really want to do is be able to look at, in detail, single molecules or single particles.
And is photo damage there typically a problem, looking at single systems?
Yeah, it's something you have to bear in mind for sure, right? A lot of the new advances in microscopy, like light sheet microscopes, one of the big advantages is you have much less photo damage. So you can follow things both, well, in 4D, right? 3D structures changing with time. For a long time, the issue with that was just so much photo damage, photo bleaching, that it was very hard to do. So now these new technologies help a lot with that.
Is there anything else we have
that we
missed
haven't
we'd
covered?
like to share?
Like one thing that you would want every person to know about your field of research, where you...
I just think it's such a fun and interesting field, right? We're always finding new things, new unexpected things come up all the time. So it's just so exciting and interesting, and it's really a great privilege to be able to work in this field and really study something so fundamental and come up with crazy new physical, chemical phenomena all the time. It's really great.
Thank you very much.
Yeah,
Okay.
thank you for the great
Yes.
conversation.
Okay, thank you.